And our next speaker is Betsy Elizabeth Komives, who is going to talk about the role of intrinsic disorder in NF-kappa B signaling.
Well, I couldn't have asked for a better talk to precede mine for introducing the whole concept of how IDPs switch binding. This was just wonderful that Peter introduced all the topics that I'm going to be talking about today. But I'll be talking about them in a slightly different transcription factor regulation system than the one that Peter talked about. And I'll be telling you about the role of intrinsic disorder in NF-kappa B signaling. And what got me interested in this system, maybe 15 years ago now, was this pretty little picture that one of my colleagues had on his wall of the partial crystal structure of NF-kappa B. These are the dimerization domains and one N-terminal domain of NF-kappa B. And it's bound to this inhibitor, which is anchor and repeat protein I-kappa B alpha. And I don't know, I think biology is so beautiful, especially protein structures. So this structure really struck me as being very pretty. So NF-kappa B is a stress response transcription factor, just like the HIF1 alpha that Peter was talking about just now. And so when a cell is confronted with various types of onslaughts of attacks, like infection or growth regulators, cytokines that turn on inflammation. The NF-kappa B transcription factor is turned on, and hundreds of genes are activated for cell survival, inflammation, cell proliferation. And so just like in the HIF1 alpha case, we have to think about how is this system controlled, because you really don't want to have all this going on constantly. And in fact, we know now that stress really does a lot of things to people's body, and chronic inflammation also does a lot of things to mess up people's normal biology. So we have to be very careful how to turn this on and turn this off only when it's needed, and not have a system that somehow leaks into this transcription activation program. So the words NF-kappa B and I-kappa B actually denote families of proteins, not just a single protein, although today what I'm going to tell you about is a single pair of proteins. So the canonical NF-kappa B, when people use this term generically, what they're talking about is a heterodimer between this protein that's called P65 or REL-A and its binding partner P50. So we're going to have a heterodimeric transcription factor between these two proteins. And then the inhibitor that I'll tell you about today is I-kappa B alpha. That's the one with this anchor and repeat domain that you saw on the very first slide. And these inhibitors of NF-kappa Bs are called I-kappa Bs. So just as what Peter was showing you before, this transcription system is very rapidly turned on when stress hits the cell. All the NF-kappa B goes to the nucleus from the cytoplasm, and then it's rapidly removed from the nucleus. And our question is, how is this system going, how is this protein going in and out of the nucleus so quickly? And Alex Hoffman, a long time ago now, showed that it's I-kappa B alpha that is regulating the NF-kappa B presence in the nucleus or absence in the nucleus. And you can see that the I-kappa B alpha is here when the NF-kappa B is not in the nucleus, and it's not here when NF-kappa B is in the nucleus. So now this system is fairly well understood that what happens is all of the NF-kappa B is kept in the cytoplasm, bound to the inhibitor. This is an extremely tight complex, about 40 picomolar is the binding affinity. And you require an extracellular signal to turn on a kinase that phosphorylates the disordered N-terminus of the I-kappa B alpha, which then targets it for ubiquitination, and the proteasome actually eats the I-kappa B off of the NF-kappa B, and that exposes the nuclear localization signal, allowing the NF-kappa B to go into the nucleus. And just exactly as Peter showed you for the HIF1 alpha, one of the genes that is turned on by NF-kappa B is the gene for I-kappa B alpha. And so even though we degraded all of the I-kappa B alpha in this signal turn on step, now we make new I-kappa B alpha that is responsible for shutting the system down. And we had structures to go by in our beginnings of this study. We knew the structure of NF-kappa B bound to DNA from Gori Ghosh's lab, and we also knew two structures of the inhibited NF-kappa B, one from Steve Harrison's lab and one from Gori Ghosh's lab. And I would like to point out right now that when you look at these structures, you see that the DNA binding occurs between these two dimerization domains and these two N-terminal domains, whereas the inhibitor binds mainly to the surface, the dimerization interface of the dimerization domains, and there's actually this disordered tail of the inhibitor that's sort of laying in the DNA binding cleft. So here's the outline of my talk finally. I'm going to tell you about how free I-kappa B alpha is, has disordered regions in the ankyrin repeats, and that this allows it to be degraded quickly in the free state and stabilized in the NF-kappa B bound state. I'm also going to tell you about how I-kappa B alpha affects the NF-kappa B binding to DNA. And then I want to get into what's going on in the NF-kappa B, how it's disordered in the free state, and how it changes its order in DNA bound or I-kappa B alpha bound. So most of what I'm going to tell you about today is work that we've done with an approach that I've worked on a lot in my lab, amide hydrogen deuterium exchange. So this is a very old slide from the old days, but I wanted to bring up the point that when we do these experiments, basically what we're doing is we're taking a protein or a protein complex and we're sampling which amides are actually being exposed to solvent transiently on a short time scale. So we're looking at deuterium exchange times of 0 to 5 minutes. So these are amides that are not well structured. I don't know if you're familiar with amide exchange, but typically amides on the surface of a protein will exchange within seconds. And if a protein is very well folded, some of the amides in the interior of that well folded protein may not exchange for months. So there's a huge time scale that we can sample. Now these rates, it's very tricky to understand the rates of amide exchange because they depend on a transient opening and closing. And when that transient opening occurs, can that amide exchange before it closes again? So I really think that amide exchange is a wonderful way to detect intrinsically disordered regions of proteins because they will exchange their amides very quickly. And this is exactly what I'm showing you here. This is actually early data we collected on I-kappa B alpha, where some of the regions of I-kappa B alpha don't exchange their amides at all, and others of them exchange most of their amides very rapidly. So when we did these experiments on I-kappa B alpha, what we found was that there are regions of the 5th and 6th ankyrin repeats that exchange. This is ankyrin repeat 5, different segments of ankyrin repeat 5 that show exchange of all their amides, and the same with ankyrin repeat 6. All of the amides are exchanging. when this free I kappa B alpha is bound to NF kappa B, now we see that those amides are no longer exchanging. And you can see this is a difference of 6 amides, 8 amides. These are huge differences in amide exchange. That I kappa B alpha seems to be exposing its amides in the free state, but locking down in the bound state. And just to remind you, we know from cell biology experiments that that bound state, which is not exchanging its amides, has a half-life in the cell of over 48 hours. It's extremely tightly bound. Whereas the free protein has a half-life of less than 7 minutes. It's very short-lived. So we played around a little bit with mutations in I kappa B alpha, and we found this mutant. It's 2 mutations in the 6th repeat, where actually the protein is degraded more slowly in cells. This is the degradation in cells. And this protein also doesn't exchange the amides in anchor and repeats 5 and 6 as rapidly as the free protein, the free wild type protein. Oops. And what I want to tell you is that that this region right here in anchor and repeat 6 is actually a degron. So it turns out, I don't know if you're familiar with this nomenclature, but a degron is a small sequence in a protein that will target it to the proteasome for degradation in the absence of ubiquitination. And so it turns out that I kappa B alpha is degraded quickly in the free state because that degron is being exposed. But in the bound state, it's not. And that's the root of this degradation difference. So let me continue on with this idea of this anchor and repeats 5 and 6 being disordered. We really wanted to try to understand, I'll just say that there are many different ways to study intrinsically disordered proteins, and one of my sort of mantras in life, I guess, is the more experiments you can do on a system, the more you can learn about that system. One of the experiments that we had done early on with I kappa B, NF kappa B complexes was circular dichroism. And what we had found, which was very puzzling to us, was that even though all the amides were exchanging in anchor and repeats 5 and 6, when we compared the circular dichroism spectrum of I kappa B alpha, these are all these helices here, we should have seen more helical content in the bound state as compared to the free state if this part of the protein was disordered, right? But we didn't. We didn't. The subtracted circular dichroism spectrum showed no change in helical content. So that made us think, okay, how can we think about this disordered region then that has its secondary structure, but at the same time, it's exchanging all its amides? So what we did was single molecule FRET. We put a donor and acceptor fluorophore in different repeats of the I kappa B alpha. We attached this protein to a microscope slide, and we watched the FRET, the fluorescence resonance energy transfer, between the donor and the acceptor fluorophore. And what we saw was that when I kappa B alpha is bound to NF kappa B, we get a very nice distribution of high FRET signal. The donor and the acceptor are perfectly positioned next to each other. Nothing's funny about this. We measured the distance in the crystal structure, we put the fluorophores where we knew they should be, and we got the FRET we expected. But in the free state, we had this tail on the fret distribution going to lower fret. If we added a little bit of urea, the tail got bigger. That mutant I told you about didn't show the tail. Unless we cranked up the temperature, then we started to see a little bit of the tail. And what was nice about these single molecule FRET experiments, we did them by turf microscopy. So we attached this protein to the microscope slide and we could see it. We could watch it in real time. And what we saw was that this is not actually a separate state. This is an ensemble of stochastic moving parts where we have high fret for a while, and then the protein will sort of move open, move closed. Open, close, open, close, open, close, open, close. Open, close, open, close, right? It's completely stochastic, and the time scale is very long. These are seconds time scales. When we bind it to something, where's the bound one? Here. Doesn't do that anymore. So now what we understand is that's why the free protein is degraded. It's not that there's no helix there. It's that the protein loses its structure for some transient amount of time. And when it loses that structure, then the degron is exposed. These are the degron data. It's kind of amazing. These degrons, I think, are very interesting, little sequences. If you take the little piece of sequence from anchor and repeat 6 and put it on GFP, that's a very stable protein. All of a sudden, the GFP is all degraded. This is GFP without that little sequence from AR6, and this is GFP with that little sequence from AR6 attached to it, and it's just disappeared from the cell within 5 minutes. So it only takes a few little openings of this structure in the free protein to have free I-kappa B alpha completely degraded. Now, it's amazing because this is one of the puzzles to me in biology. Over 90% of all the I-kappa B alpha we ever make is just degraded. It never manages to find an NF-kappa B, and it's gone. The free I-kappa B alpha has to find an NF-kappa B, and then it will be degraded through the signaling of the kinase, et cetera, et cetera. But if it doesn't find an NF-kappa B, it's just degraded. And over 90% of our I-kappa B that we make is just degraded. At the end, I'll tell you why I think that's important. Okay. So now, I want to tell you about how I-kappa B alpha affects NF-kappa B binding to DNA. So we showed a few years ago, this is now following up on what Peter Wright was talking about earlier, that that I-kappa B alpha actually facilitates the dissociation of NF-kappa B from DNA. And we were able to observe the formation of this transient ternary complex in the stop flow experiments before the NF-kappa B dissociated from the DNA. And the more concentration of I-kappa B we add, the faster that dissociation occurs. And it's always much faster than the free DNA dissociation in the absence of I-kappa B alpha. And we measured all the kinetic rates, and just as Peter was telling you, this process is essentially irreversible. Once you form this transient ternary complex of the NF-kappa B, the DNA, and the I-kappa B alpha, there's no going back. DNA dissociates, and you get this very stable NF-kappa B, I-kappa B complex. So we wanted to probe this further, and one of my students, Holly Dembinski, noticed that at the very C-terminus of I-kappa B alpha is a highly acidic sequence. We were trying to figure out how does this dissociation of the DNA occur. So she decided that she was going to mutate all of the negatively charged amino acids in this C-terminal sequence to neutrals. And we found that the KD for this I-kappa B alpha was only 4-fold lower than for wild type. Now, when it's 40 picomolar and you multiply that by 4, you're still very tightly bound, and that's key. But when she took that protein and tried to do the stop flow experiments, she found that instead of dissociating the DNA, she ended up with a different signal where it looked like the DNA was still at least partially bound. And what we ended up determining through fluorescence anisotropy experiments is exactly that the wild type protein binds and forms a ternary complex with the DNA, but the DNA really doesn't want to stay bound there. And so the equilibrium between the DNA bound and DNA free is in favor of the DNA being unbound and the I kappa B being bound to the NF kappa B. On the other hand, in this mutant, where we have no negative repulsion with the DNA doesn't really mind being there so much, and we have more of an equilibrium favoring this ternary complex. Now, this is not a stable ternary complex. You cannot put this thing on a size exclusion column and have it come out with the DNA still bound. So there was no way we could do structures of this thing. So again, we turned to hydrogen deuterium exchange. We were able to add enough DNA to these experiments to probe what all those amides were doing in the binary and ternary complexes, NF kappa B, I kappa B versus NF kappa B, I kappa B with DNA bound in this mutant. And what we saw was all throughout regions of there was one segment of ankyrin 3, and then a segment of ankyrin 4, and then a lot in ankyrin 5 and ankyrin 6. The pink dots are the ones you have to focus on here. What we saw was that here, let me just move on to here so you can see it on the structure. What we saw was in the ternary complex, when the DNA is bound, the I kappa B alpha adjusts itself, if you will. So, and we had proposed this way back in the day, and I was so happy to see these results. We had proposed that the I kappa B alpha folds on binding to NF kappa B, and we had said that based on that dramatic decrease in amide exchange in these ankyrin repeats 5 and 6. But we know that the folding of I kappa B alpha actually requires subtle interactions between all of these ankyrin repeats throughout the whole protein. And that's exactly what we saw in this ternary complex. The chemical shifts changed and the amide exchange increased when the DNA was there all the way throughout this I kappa B alpha, as if it couldn't really attain its fully bound structure when the DNA was still there. It got part of the way there, but not all the way there. So we turned this, we caught I kappa B alpha in the act of stripping the DNA off the NF kappa B. And the other result that I really wanted to show you is, because this is super important to me, just because we see something sort of interesting in proteins, does that really translate to something important in the cells? Oh my gosh, okay. I said I was gonna be on time, and here we are. So what we saw was when we incorporated the 5X pest mutant I kappa B alpha behind the normal I kappa B promoter in cells, we saw that I kappa B alpha cannot bring the NF kappa B back out of the nucleus. So normally, I showed you at the beginning that when I kappa B is synthesized after NF kappa B goes into the nucleus, it brings the NF kappa B back out of the nucleus. Well, that mutant is much slower in bringing the NF kappa B out of the nucleus. So that tells us that this stripping rate, taking the NF kappa B off the DNA and bringing it back out of the nucleus, is what actually controls that nuclear export of the NF kappa B, so that we turn this system off, we don't signal anymore. And through computational studies that we've done in collaboration with Peter Wallenus's lab, what we now know is when the I kappa B alpha binds to the NF kappa B, it doesn't just electrostatically repel the DNA, but it also twists the DNA binding domains to prevent DNA binding. So this is also allostery going on here. I am going to rush through the rest of this. Okay, so NF kappa B. is predicted by the IUPRED score to be highly disordered in this N-terminal binding domain. And this is the domain I showed you the crystal structure of at the beginning of my talk. So now we know that that was the crystal structure of the DNA-bound form. But actually, when DNA is not bound to NF-kappa B, this N-terminal domain is exchanging all its amides very quickly. So it itself, this entire domain, is folding on binding to the DNA. And we can see that here. The green is DNA bound, the black is no DNA bound. Everywhere in the protein, we see decreases in amide exchange. Interestingly, up at the other end of the protein, far away from where DNA is binding, we see allostery. We see an increase in amide exchange way up here. This is the segment leading to the transcription activation domain. That's kind of interesting, isn't it? But I-kappa B, on the other hand, stabilizes these N-terminal domains even though it's not binding to them. It's not binding to them. Here's the I-kappa B. Here's all these stabilized regions down here. So there's a lot of long-range allostery going on in this system, just as Peter promised you would happen. All right, and I guess my time is up, but I will just make these remarks really. It's basically what Peter already told you, that when you have a stress response transcription system, you need to rapidly turn it on, and you need to rapidly turn it off, and you need to turn it off completely. And that's what happens when I-kappa B binds. Okay, done.
Thanks, Betsy.
Did I make it? Almost.
For the very lively presentation. We would have definitely missed something if you didn't make it here. So we have a few minutes, yeah? First question there.
Who should I go with first? The one with the microphone. Yes.
Oh, me. Cool. So the large-scale motions, you could see them by total HDX, by global HDX. So have you? And actually, why don't you see them? Why is your? Well, maybe you didn't have error bars, but on the peptide level HDX, I would expect the error bars to be really big in some of the peptides because of that large-scale motion. But why not?
Okay, I'm glad you brought this up, and I don't know how much time we have.
Okay.
The error bars are very small, but that's because we use a robot to do our experiments. And actually, this is something very interesting because when you have these weakly folded proteins, the reason you have high error bars is because your room temperature is not exactly the same every day. And if you do your experiment at 22 degrees or 24 degrees or 26 degrees, you'll see very different amounts of amide exchange and whether or not you equilibrate your sample.
No, I'm saying it because of the conformational change. No, so you have two conformational states there. So you have
no,
one.
we don't have two conformational states. That's what I was trying to emphasize in the single molecule FRET. We have stochastic openings and closings to varying extents. There is not two states. There is not two states in this system at all.
But then you have no HDX evidence for that.
You can't, because the HDX is just an equilibrium sampling at whatever temperature you're at.
Then I would expect bigger error bars. That's why I asked the error bar question. Okay, but we can talk about it.
But you'd be putting error bars on single molecules and averaging them over time. So your error can be very small on that average of the large ensemble. That's the way to think about that. I know what you're saying, but yeah.
So that was really beautiful. I noticed in your first, when you first showed us the NF-kappa B going to the nucleus and then coming back out, or at least disappearing from the nucleus, that then it went back to the nucleus and it seemed to persist there.
So what's going on there is actually that as long as you have kinase, then you get another cycle of this thing. So
does that kinase stay active, or what turns that off? It seems like there needs to be another turnoff, right?
That's exactly right. So the system basically, so basically what happens is the kinase is there at a low level. There are lots of other mechanisms of turning the kinase off, as you can well imagine. But that experiment, how should I say this? Yeah, that experiment just really sampled a pulse of TNF alpha and what happens there, which isn't what really normally happens when you have a stress response. Yeah. Another.
So we can have one more question and then we have to cut it off, sorry. Just
Okay,
one more
that one has the mic over there.
Yeah. Hey, I was curious if you could therapeutically target the degron in Kappa B
That's a brilliant idea.
to basically BNF Kappa B pathway.
Yeah, that's a great idea. I don't know how I would do that, but that's a great idea.
All right.
Okay. Get out of here.
Okay, let's thank Betsy again for this entertaining talk.